All right, let's get started. First, I introduce myself. My name is Richard Kondo, and I'm VP of Strategic Partnerships at AnaBios. And I want to thank you again for joining us for another round in our webinar series on investigations in biomedical issues and how they relate to human disease. If we could go to the next slide, please. So I will introduce AnaBios. AnaBios is a unique contract research organization and biotech based in San Diego. We are entirely focused on translational research, conducting and utilizing human tissues recovered from ethically consented donors. We have developed methods and compositions that allow us to maintain the viability and function of recovered human tissues and cells. These technologies enable the study of drug activity in human ex vivo preparations, or the investigation of pathological mechanisms of disease states in authentic human disease tissue. Over the years, we've applied these methodologies to support translational research in a variety of therapeutic areas. We have been especially active in pain research with work conducted on human dorsal root ganglia neurons. We've expanded these activities to recovering viable human spinal cord, and more recently, we've developed capabilities to recover trigeminal ganglia and whole human brain. We have 3 brochures describing the neurotissues available at AnaBios. They're listed in the handout section that's in the panel, control panel that you can see there, and you can download them from there. Also, you will note that there's a questions panel, and after the talk, you can use that panel, and I will remind you again to allow you to do to submit questions. So on to our speaker, and next slide, please. So today's speaker is Dr. Wenqin Luo. Dr. Wenqin Luo is Associate Professor of Neuroscience at the School of Medicine at the University of Pennsylvania. She received her doctorate from Johns Hopkins University and afterwards did postdoctoral research at David Ginty's lab at the Howard Hughes Medical Institute at Johns Hopkins, as well as Lawrence Katz's Howard Hughes lab at Duke University. Her current research is focused on understanding the function of the somatosensory system. She and her team explore the interplay between touch and pain and is identifying neural populations and pathways in the interconnection between spinal cord and dorsal root ganglia. Today, she will present new data, and the title of her talk is High-Depth Single Soma RNA of Human DRG Neurons. Thank you. All for you, Dr. Luo.
And can everyone hear me?
Yes.
Can people hear me well? Okay.
Yes.
So let me just change back that to laser point. Okay. Rich, thank you for a very nice introduction, and it's my great honor to present here today. And I'm very excited to talk about this new story, and also to get the feedback from the audience. Before my talk, I just want to make sure I do the acknowledgement first. First, for this project, it wouldn't be possible without the precious, sharing tissues from human donors, and we got those through the help of NDRI for human tissue procurement. We use the Penn SB DRC LCM facility for this project, and also for the NIH and the UPenn funding for making it possible. And this project is really a very nice collaboration between my lab, which is conducted by Dr. Huasen Yu, a postdoc, and also Dr. Patrick Info's lab at Karolinska Institute, and also Dr. Hakan Al-Nelson and Sid Nagi's lab at Linköping University. So I really appreciate that they gave us this opportunity to working together and to get this exciting story come together. I also want to thank my friend Yulong Li, who encouraged me to take this really brand new direction of my lab research. Okay, for this audience, probably it would only need very little introduction about the human or the mammalian somatosensory system. It's a very versatile sensory system. It can generate perception of many different modalities of sensations, pain, itch, temperature, touch, body position, and it's responsive to a variety of external and internal stimuli, and the organization of the somatosensory system allow us to generate the information of what the stimulus are and where does it come from. So this versatile function of our somatosensory system really arises from the heterogeneous primary somatosensory neurons that are located in the dorsal root ganglia and the trigeminal ganglia. So this is just an illustration to show a half cross-section of DRG and the spinal cord. So for people who have worked on this field, we know those neurons are just very different, very heterogeneous with regard to their soma size, molecular markers, their physiological property, their anatomic connections. But the question is really, I mean, for people working in this field, really how many different type of somatosensory like DRG and TG neurons do we think they are? So actually, I would say this is evolving. This is a question with evolving answers because using traditional method molecular markers, we might identify several types. But nowadays, with particularly the single-cell RNA sequencing, we might discover much more kind of molecular types. Okay. So my interest of this direction really comes from a research project we did several years ago in collaboration with Dr. Yun Li's lab that really seems like very interesting to me about this species difference, because I've been working with the mouse somatosensory system for a very long time and take it for granted, this is a mammalian system. But for this particular project, I mean, Dr. Xin Dongdong's lab also, around the same time, we kind of identified that bile acid and bilirubin might bind to the human MRGPRX4 receptor that can trigger the cross-static itch in human. And what's interesting is previously in mouse system, another GPCR called TGR5 has been identified for this function. But when we look at the TGR5 expression, it's indeed in mouse DRGs, it's expressed in the mouse DRG neurons. However, when we check both monkey and human DRGs, this TGR5 expression is in the satellite glia cells around the sensory neurons, but not in the sensory neuron itself. So here shows immunostaining, but we also have in situ hybridization data to back it up. So this data is really intriguing and make myself to start to really think more carefully about this species difference. Because if we just, smash the human DRG and do RT-PCR, we would find a TGR5 expression there. But who knows? It's not in sensory neurons, but in the satellite glia cells. So this is it's an image I adapted from the Tanks Genome Block, website, just to show briefly the mechanism of the single-cell RNA sequencing, how it works. Basically, with a mix of heterogeneous cells, you dissociate, you get individual cells, and then you will be able to sequence them to see their molecular profiles. That's, as I just talked about, it has advantage for over this bulk sequencing because now you have this cell type specific information. But when it comes to human neurons or the human DRG and TG neurons, there are certain technical hurdles or difficulties we need to consider when how to do it. So for human tissues in general, the availability of this good quality of human tissue is a pre-requirement. They still have good amount of mRNA, not degraded. Usually these tissues are dissected or in a surgery room far away from basic research labs, how to preserve the mRNA during this transportation process, and also even during the cell dissociation, the transcriptome may change. So how to preserve this more to a kind of like native endogenous status. And also there's a difficulty in isolating and enriching this human DRG TG neurons from non-neuronal cells. Only when we start to work with the human DRG, we realize there's a huge, basically in human DRG, the amount of non-neuronal cells are much more overnumbered than the neurons. So after dissociation, usually what's remaining are non-neuronal cells. And also because these cells are less in number and also they are much larger in soma size. So they are more prone to damage during enzymatic and mechanical dissociation. So it's actually very difficult to preserve their soma, their cell body after the common enzymatic or mechanical dissociation procedure. which is successfully used for mouse cells, mouse DRG neurons, TG neurons. So so far, there are 4 nice studies published along this direction for conducting the single-cell RNA sequencing of human DRG or TG neurons. Because of the technical difficulties I just mentioned, 3 of them did the single nuclei sequencing because after dissociation, if the cell body is broken, nuclei are still there. And then 1 study using the 10x VCM spatial transcriptomics, that's kind of gets the soma, but as I'm showing here, for the limitation of the current method, 10x VCM is nice. It gets the soma transcript, but this 50 micron capturing dots, if we think about, because the human DRG neuron soma size varies from like 15 to 110 micron almost, so the chance for captured dots to perfectly match to a neuronal soma, the chance wouldn't be very high. So most of those is a mix of neurons and some of the non-neuronal cells. And then for the single nuclei sequencing, because of the nature here, here just 2 RNA-scopic images my lab did just for 2 genes. And these are 2 single neurons, and the black hole in the middle are the nucleus. So it's just the nature, both the quantity of the mRNA, it's mainly enriched in the cytosol, in the soma, and also if we think about the quality, nucleus likely contains the nascent mRNA, so the transcriptome species from the nucleus to cytosol may also be somewhat different. So here I'm just showing 1 example of the single nuclei sequencing. The first one did, and because of that, maybe because of that reason, then MRGPRX1, which is we know it's a pretty well-known GPCR for each receptor expressing human DRG neurons, actually are not well detected by the sequencing dataset. So because of that, when we started this project, we wondering it would be nice if we could get the soma transcript, but also maintain the single cell resolution. So the question is, can we do that? So this is what Hua-Sen did. He developed a novel method for doing that. So NDRI helped us to procure the human DRG tissue. I will show that information in the next slides. And these human DRGs are embedded in OCT immediately for mRNA preservation. And those blocks were sent to my lab in freezing, so a frozen box, again, to preserve the mRNA. And Hua-Sen would cryosection them to the slide, and then we will bring to the LCM facility with a brief staining. Here we can see nicely those large human DRG neuron cell bodies, and the laser can cut this single cell, and that falls to the cap. And then after that, Hua-Sen used the Smart-seq2 protocol. So that's a more deep sequencing protocol compared to 10x genomics, which allows us to, again, get more kind of transcript and then sequencing and sequencing data analysis. So here are the donor and the DRG information. We so far have worked with DRGs from 3 donors. This is their age, their DRG levels. So we sequenced the 60 human DRGs. They are at the lower thoracic and lumbar level. So those are the DRGs governing the sensation from our lower back and from the legs. And we did this sequencing from 3 batch. And total, we sequenced more than 1,000 human DRG neurons using this approach. So here, so after the initial data analysis, this is in together with Patrick Info's lab, we found about 10% of those human DRG neurons has kind of obvious like satellite glia cell contamination. So those cells were taken out, removed for the data analysis. So now we have about 1,066 DRG neurons for all the remaining analysis. And on average, we have more than 9,000 unique transcripts per soma. So if you remember from the previous table for the single nuclei sequencing, that's usually about 2,000 or less. So our sequence, the transcript, the unique transcript we get from this method, basically is much, much more. And we don't see a clear, we see a comparable quality from 3 different batch, different donor, or different DRG levels. And also, if we compare the soma size distribution of all DRG neurons to those dissected DRG neurons, we see very similar soma size distribution. So that's suggesting we don't have obviously sample bias here. And so this 1,066 human DRG neurons are clustered into 16 different clusters. So here I'm showing the results using the Surat cluster, but we also validated this clustering using 3 different methods. We did kernels, we did machine learning. All these different methods shows very similar clustering. So that's really shows basically we are very confident in how robust the clustering has been. And as you can see from here, for all those 16 clusters, they all express neuronal specific markers in synaptophysin and also PGP 9.5. And so who are those 16 different clusters, right? So we first look some of the gross features of those clusters. So first we look at their soma diameter and the expression of neurofilament H. And if you look at that, we see a really interesting division here from cluster 1 to 7, seems to have smaller neuronal diameter and also very low expression of neurofilament H. But from 8 to 16, they have higher level expression of neurofilament H and also larger soma size. So it seems likely 1 to 7 more like C fibers, 8 to 16 more like A fibers, if we think about it. And then if we look at another gene expression pattern of another, human pain disease gene, PRDM12, for neurodevelopment, we see PRDM12 shows some obvious expression from 1 to 12, but not in 13 to 16. So that makes us think maybe from 1 to 12 are those C and A fibers more related to kind of human pain sensation, but 13 to 16 are more likely to be those kind of low A fiber, low threshold mechanoreceptors. That's not their primary function is not related to pain sensation. And then lastly, if we look at the PEP marker, CGRP, we actually found there are 8 types of CGRP positive human DRG neurons in our data, and that division would further separate the C fibers into non-PEP fibers 1 to 4, and then the PEP C fibers. So these are our kind of like the gross features of the 16 different human DRG neurons. So as I just said, then that would allow us to say maybe 1 to 4 are likely to be non-pubertergic C fibers, and then 5 to 7 are pubertergic C fibers, 8 to 12 are pubertergic A fibers, and then the remaining are likely ALTMRs. So now it comes to different types. So based on their top molecular marker expression and their molecular profiles, so this is how we give their name, and we kind of speculate or think about their potential functions. So the first type is, so the way we name the neuron H means the human, right? And then for the non-pubertergic, non-CGRP positive fibers, we kind of used the monoclinature system from the mouse system. So it's just easier for people to understand and also correlate their function. Such as TRPM8, CLTMR, MP1, MP2, or A delta MT1, A beta LTMR. So that's basically how we name it. But for the CGRP positive neurons, because it's just very different from what we saw from the mouse system. So how we name it is we have human PEP, that means they are CGRP positive population pubertergic with a dots, and then with a top either molecular marker or functional marker indicated here. So basically, the H-TRPM8 is a small population that express, TRPM8 with for photosensation, and then this is CLTMR here, the MP1 and MP2 here, and then from here is the PEP, somatostatin, all the way to here are the CGRP positive neurons, and the PEP0 is a strange CGRP population that they express CGRP, but we don't find any other distinguishing marker. So that's the reason why we call it PP0. And on top of here are the A fiber low threshold mechanoreceptors. So Patrick's lab did cross-species comparison of our human DRG neurons, the single soma data, to with the mouse single cell data. So this is from the schema data from the Ginty lab, and then the macaque data that generated by Patrick's lab. So we, again, for this cross-species comparison, we did multiple methods. We did Suros, we did Kanos, we did machine learning, multiple methods. And I think the conclusion is fairly similar. So I would just show some, just see some gross conclusion here. So generally saying, we see a more relative conservation. So here, if you see a T shape, that means really very well conserved. For example, the human CLTMR and the mouse CLTMR seems from the molecular profile are very well conserved, and so does that to monkey. So generally saying, this kind of non-prototypic fibers are more evolutionary conserved from mouse or between human and mouse or human and macaque. However, for the nociceptors, for the CGRP positive nociceptors, we see much more great divergence. For example, for the mouse alpha, beta, gamma, or even gamma, first three, like the mouse CGRP positive neurons, actually we don't find any of those in human. And then, as I'm going to go to detail later for different populations, for certain populations, PEP populations, we also don't find good correspondence in mouse or macaque. So suggests they could be human-specific populations. So basically, from this cross-species comparison, I think the general impression is the CGRP positive nociceptors display much more evolutionary diversity, while the non-CGRP positive sensory afferent shows more conservation. Patrick's lab also did this transcriptional factor associated gene regulatory network analysis. So here the black one is the human and the macaque and mouse, and the yellow outlined programs are those conserved in all three species, and the green outlined programs are present only in one species, or like here, like primate between human and monkey. So if we, even in those kind of like conserved cell types, if we look at their molecular profiles, there are really a lot of molecular profile difference. So here, just showing one example. So here, Watson plotted the top 10 marker genes of each human population, and then to plot them in the monkey and mouse data. So for example, for the human TRPM8 population, it relatively conserved across the species. However, if we look at the top one marker gene for the human TRPM8, which is called STUM, this gene is more broadly expressed in monkey neurons, and it's not expressed in mouse neuron at all, mouse DRG neuron at all. And then the same thing is for the human CLTMR population. Their top marker in human is the CASQ2. Again, this gene is barely expressed, is barely detected, let's say, in the current macaque and mouse single-cell RNA sequencing data. And then the same thing is true for the human MP neurons. For the MP2, for example, MRGPX3 and MRGPX4, those two are the primate-specific gene. It's not expressed in mouse, and even in monkey data, they seem are more widely expressed than this really sharp expression in the human MP2 population. And more examples for this human PEP KIT population. The top marker KIT, which is a receptor tyrosine kinase, again, it's more broadly expressed. It's much more broadly expressed in both monkey and mouse data. And then for this PEP CRNA7 population, so this is a population correspond to macaque PEP2 population. So cell type wise, this is relatively conserved. However, this molecular marker, top molecular marker, ChRNA7, seems specific for human. It's not expressed in the mouse data at all. There are more examples, for example, for the A delta LTMR, then for this DCHS2 genes, it's primate specific, it's in human and monkey, A beta ALTMRs, but it's not, again, it's not expressed in the mouse data. So, okay, so next I'm just going to go through each of those population one by one. to validate some of the top molecular markers and also discuss our speculation about their functions. So the first 4 population we discussed are those non-proteolytic C fibers. The first one is TRPM8 population, and they highly express this cold and menthol receptor TRPM8, as we can see here. They also express another mark gene, STUM, as I just said, which seems very specific for the human DRG neuron. And this is double RNAseq in situ and their quantification. So based on this high expression of TRPM8 and known TRPM8 function for cold temperature and menthol, we think this seems like a designated population for temperature and chemical sensation. And then the next population is this CLTMR. It's expressed, again, a human-specific marker, CSQ2. It's expressed a relatively high level of piezo2, not surprising, as a low threshold mechanoreceptors. And this is on a scope to show their co-expression. And based on previous results pioneered by Harkin's lab, we think this population will mediate, this pleasant aspect of tactile sensation. And then the next 2 population, and the human MP1 and MP2 population, they are really marked by this very high and specific expression of MRGPRX1, the G protein coupled receptor for chloroquine and all of the other itch-related ligand. So what's different between the MP1 and MP2 is MP1 express a relatively higher level of piezo2, like one feature, than MP2. So that's kind of a little bit similar to mouse MP1 versus MP2, because mouse MP1 is marked by MRGD and they are mechanosensitive, slightly maybe more mechanosensitive. And they also, if we look at numbers, if you remember, in humans also MP1 from our presentation, MP1 has much larger number than MP2. So these 2 populations also express histamine receptor, interleukin-31 receptor, and co-receptors, and many other actually each receptor. So based on those, we think these 2 populations are also like for the itch sensing population. And then the next 3 populations are proteolytic C fibers. And then the first one is the HPP somatostatin population. So this population in mouse and monkey previously are called the MP3, the non-proteolytic type 3. But here we call it PP somatostatin. The reason is because based on our sequencing data in human DRG neurons, we detected relatively high CGRP expression in this somatostatin population. And here is the RNAseq in situ. And this population shows this really beautiful, unique expression of somatostatin. They also express histamine receptors and interleukin-31 receptors and co-receptors. So this is, again, another likely itch sensing population. We think they function especially maybe during inflammation. And then the next 2 populations, they are marked by their overlap expression of TRPV1. So this 2 population and TRPA1. So these 2 populations shows high and overlap expression of TRPV1 and TRPA1. We call them 1.1 and 1.2. So they are a set of molecular markers that basically why these 2 are clustered, are separated. One shown here is PTGR3, but we also have other markers. So basically, based on those markers, because some of those were found from the mouse retrograde labeling, those markers are more expressed in deep innervating afferents. So based on those scattered knowledge evidence, we speculate that the PP TRPV1 A1.1 are the chemical, like polymodal nocitive fibers innervating the skin. It's mainly for temperature and chemical sensation. But then the 1.2 population likely innervating deep tissues, and they are polymodal. And then the next, actually 5 populations are the peptidic A fibers. We don't discuss about PP.0 here because we didn't find any particular molecular marker for this population, even though it's a large makes a large percentage of neurons of this CGRP population. So the first one, which is very interesting, is this PEP piezo high population. So this H means high here. So this population expressed a very high piezo2, almost comparable to the A delta LTMR, which is the most mechanical sensitive fibers. So it's a little bit, intriguing why a PEP fiber need to express such a high level of piezo2. They also express high level of PTGR3, but no AAA1, which is different from the previous one. And in addition, as you can see from the additional expression marker here, this is also a rare population, also seems to have some expression of piezo1. Usually the overall expression of piezo1 in human DRG neurons is very, very low, but this population seems to have a little bit, maybe. And then we also found a very unique expression for adrenergic receptors. And this receptor previously was shown to be a specific marker for sensory afferents innervating human artery, or the artery vein, like the structure. It's also expressed PTGIR, again from the mouse retrograde labeling study. This seems come from bladder. So from all this kind of different scattered knowledge and also the molecular profile, we actually want to propose this is a population that innervating the blood vessel for sensing blood pressure and regulate local blood flow, or innervate bladder for the bladder mechanical sensation. Given the body size difference between mouse to human, actually this requirement to sensing the local blood flow and regulate to sensing the local blood pressure and regulate blood flow actually is a much huge problem. Like it's not it's different magnitude basically if we think about this problem in human body than the mouse. And we think this may be the reason why either this population is kind of like human specific, because we really do not see those CGRP such a high piezo neuron in human data, in mouse data before, or it could be greatly expanded cell type in humans. And then the remaining three type, one is this PP kit. As I mentioned, it's marked by this high expression and a specific expression of this receptor tyrosine kinase kit. It has some expression of piezo2, but not as high as this piezo high population. This population is similar to monkey PP3. So I will talk about this population more later. And then this CHRN7, again, as I said, this molecular marker is human DRG neuron specific. They are more similar to the monkey PEP2, even though this molecular marker is unique, it's different. And then for this NTRK3 population, they highly express NTRK3 and S100A4. This, again, it seems like a more human-specific population because we couldn't find a very close correspondent population, either monkey or mouse. And we are not clear about what their functions are. And we don't know what's the function of PP0 either. We don't have speculation. So PP kit, as I will discuss later, we think these are the population for mediate the hair pulling pain in the skin, and CHRI7 are likely P1, again, innervating deep tissue, visceral deep tissue for pain sensation. Okay, so now we come back to this the 16 different clusters. As we see, this CGRP population, the PP population in human DRG neurons seems really complicated, and maybe some of those are likely, to be human specific or new populations. As I said, I'm very curious of this PP0. It's a large population of cells, but we don't find distinguished marker, or we don't really have a good speculation about their functions. Okay, so then the last set are the ALTMRs and this human ATF3 population. So here for the A delta LTMR, it's marked by its really high expression of NKR2 and also piezo2, I showed you before. And it also has a more specific marker, a potassium channel, KCNV1 here. I would say we name it A delta LTMR But our collaborator, Dr. O. Nelson and Nagi's lab, actually from their physiological recordings, they actually was not able to record LTMRs in the A delta range, conduction velocity range. They never found such fibers. So I would say this name more reflect its molecular feature nature instead of really implicate that their conduction velocity are in the A delta range. Let me just clarify this. And then the next population is the A beta LTMR, which marked by the overlap expression of NKRK3 and NKR2, and the proprioceptor are mainly marked by this albumin expression. So as you can see, based on the current knowledge, we know for the A beta LTMR, there are I1, I2, SA1, SA2, or for proprioceptor, there's also multiple subtypes. As you see, our data currently have not been able to resolve those clusters yet due to the limited number. So if we can sequence more, I am confident we will be probably find those molecular types for each of those subtypes. Okay, so the last type is this human ATF3. They are marked by this really specific expression of neural like injury marker ATF3 and a set of like neuropeptide. So this population was first described by Nikhil's data, the Ginty's single cell RNA sequencing mouse data. They call it an unknown population. So we find this population is very similar, highly conserved to what they found there. So some people might consider, whether this is a dissociation artifact or whatever, but for our method, since we are using frozen section, we never dissociate the cell, so we are very confident the expression of those markers are not a dissociation artifact, they are there. So, and also as shown by our RNAscope in such hybridization. So we think they are a unique cluster. The reason right now is unclear whether this is a degenerating population or this is, have some function. Okay, so we find many interesting, again, as I said, molecular profiles, Watson plotted, if you see the distribution of, different GPCRs, ion channels, chemical receptors, neuropeptide, they could be potential, biomarkers, drug target, some of them show, cell type specific expressions. And here I just using one example because Huasen and we are very interested at this itch sensation. So panel A shows the itch receptors that previously has been studied, and we know those are itch receptors. And indeed, our sequencing data shows very specific expression of those receptors in this 3 population of potential itch sensing fibers, MP1, MP2, and the somatosensory. And then panel B shows the receptors that are previously thought the maybe itch receptors or functions in itch sensation or used as a drug target for treating chronic itch. But our sequence data shows none of those receptors actually are expressed in those itch sensing neurons. So they could function through other mechanisms or maybe non-neuronal cells or whatever. And then lastly, panel C are the receptors that could have not been, their function has not been clearly studied in itch sensation, but they have this really beautiful, unique expression pattern, and those could be some novel receptors for itch sensation. Okay, so so what's interesting from this sequencing data is now when we look at the sequencing data, actually we now can predict some novel physiological properties of those human afferents that previously has not been shown by using animal study or using previous recordings, basically. So one such population is this PP KIT population I mentioned, right? So it's marked by this high expression specific of KIT. If we look at, they correspond to mouse CGRP, this population. And if you look at the KIT in mouse data, it's more dispersed in several populations, but it's still highest in the CKIT population. And work from Alex Chastra's lab really shows very nicely this is a unique CGRP population in a way that they are CGRP, real CGRP population, that they are CGRP positive, but TRPV1 negative. Because we usually see a very nice, overlap between CGRP and TRPV1, but this is a real population. They are CGRP positive, but TRPV1 negative. they also express piezo2 for their mechanical sensitivity for the hair pulling pain. And in human population, the PEP kid, we see they are KIT positive, they are CGRP positive, they express piezo2, so very similar to mouse, and they are TRPV1 negative. Again, a very rare case if we look at the CGRP PEP fibers. So we are pretty sure this KIT, HPEP KIT population is, correspondence to this mouse hair pulling pain population. But what's interesting is, in human data, we see the expression of TRPM8 in this population. So this is the only other population shows this very, I would say, decent expression of TRPM8 other than the TRPM8 population itself. But this TRPM8 expression is not there in the mouse data. So what that means is, if that is the case, that would say the human PEP-KIT population, which likely to be afferent high threshold mechanoreceptors, would have cold sensitivity, cold or mensal cold sensitivity. Is that indeed the case? So here again, it's only scope to show the expression of TRPM8 in this population. So this is what Hawken and Sid did in their lab. So this is just shows the human in vivo microneurographic recordings from the human hands and legs where the units were recorded from. And this is a general cluster about their conduction velocities and their mechanical threshold. So, and then their conduction velocity here. So now, so this is a unit recorded from the human leg. This is likely to be a PEP-KIT population because based on conduction velocity, those are 8 fibers. And based on their mechanical sensitivity, they are not responsive to soft brushing, but they are responsive to more stronger mechanical force. So they are 8-fiber high threshold mechanoreceptors recording from the leg. And if that's the case, and then they are not responsive to heating stimuli, such as they are TRPV1 negative, right? So fit with the molecular profiles we just talked about. The question is, are they cooling sensitive? And the answer is yes. When they cool the skin, this neuron start to fire. So this is really a novel physiological property that was not previously demonstrated by the animal studies. And then the other population we studied is the human CHTMR. So for the human CHTMR, and also actually for mouse, it's very well known the CLTMR are also cold sensitive. However, from the expression data, as we can see, they don't express TRPM8 at all. It's just completely zero. There's no TRPM8 in CLTMR. I believe it's also the case for the mouse data. So this is only scope in situ just to show there's no TRPM8 in this population. However, what's interesting is we found from human data, this population actually expressed TRPV1. So they also express piezo2, obviously, and then suggest the human CLTMR probably would have heat sensitivity, and they should be sensitive to capsaicin, even though since they do not express TRPM8, even if they have cold sensitivity, they shouldn't be responsive to mensal. So is that the case? So this is a HCLTMR unit they recorded from the human hand based on the conduction velocity, and again their mechanical responsiveness. They are in the C fiber range, responsive to the soft brushing, so it's like fits the criteria for CLTMRs. And indeed, they responsive to heating and they responsive to capsaicin, consistent with our sequencing data showing the TRPV1 positive. And they responsive to cooling, but not mensal. So again, this is a cold sensitivity, but it's not a sensitivity to mensal because they do not express TRPM8. And the mensal works because those are human subjects. They can report when mensal is applied, they have a perception of cold, and also another background C fibers shows firing responsiveness to mensal. So it's interesting here. So the question is, are this CLTMR express a novel unknown cold receptors here, or could TRPV1 do the job? Because except that 2 population of V1A1 double positive ones, we find CLTMR actually also have some decent expression of TRPV1. So could A1 do the job here? I think those are interesting functional studies in the future. So anyway, in summary, we have developed a novel method for conducting single soma deep RNA sequencing of human DRG neurons. We believe this technique, we believe actually not just for human DRG neurons, as far as we are aware, we think this is probably the first single soma RNA sequencing for all human neurons. And from this study, from the current sequence of the cells, we identified 16 different types of human DRG neurons, but we do anticipate this number to be much higher with more neurons are sequenced. From the cross-species comparison, the CGRP positive PP nociceptors show great divergence during evolution, but the other populations, at least at the cell type wise, seems more conserved. And as I showed by multiple examples, our data set really provide novel insight into molecular and cellular mechanisms underlying human somatosensation, and also new understanding for the drug effect or pharmacological effect. And we are very, very excited that our results shows a very close relationship between the electrophysiology defined human sensory afferent types and the molecular types reviewed by our data set. So that really highlight the precising of our data set and the unique utility of our data set in molecular discoveries. So we continue collaborating with our collaborators to work on more additional those sensory afferents to try to work out this relationship. For future directions, there are many. Some of those are obvious. Obviously, I need to get the funding. We want to sequence more different levels and TGs. We want to do more physiological validation, and also it would be nice if some of the antibody works, to staining from the human tissues for histological validations. I think our method should be readily applicable for other periphery ganglia and all CNS neurons, human CNS neurons. We also would like to sequence samples from pathological conditions like chronic pain, for example. And also we are currently trying, or at least want to integrate with other single cell techniques. For example, if we can integrate ours with 10x VCM and do the cell type deconvolution, then we should be able to get both neurons and non-neuronal transcriptomes simultaneously. So that would be very, very powerful. And that's all, actually. Thanks for your attention. I would be happy to answer questions.
Thank you, Dr. Luo, for a fascinating talk on this molecular distinctions of the population. So we've already have questions coming in on the questions panel, and just reiterate on your panels, you should see a questions panel that you can type in your question. So I'm going to start with the first question, which is a technical one, which is which antibody did you use for the GPRX1 results?
So we didn't use any antibody. All images I'm showing here are on a scope fluorescent in situ hybridization.
Right. Okay. Thank you. So for a second question is, can you comment on how similar the number of genes detected are between different subtypes, and whether there might be more glial contamination in small diameter neurons than larger?
I think this is a very interesting question. So for the first part of the question, I can see I don't think we necessarily see a transcript number difference across different types. I think that number is relatively similar. So with regard to whether we got more glial cell contamination from small diameter DRG neurons, that's not my impression, actually. So we took out that about 100 cells is 10% of the cells at the very beginning, basically, after the initial analysis. It doesn't seem to us, at least not our impression, that it has any particular pattern, that contamination. So it's more, it seems more random.
Thank you. So the third question is, even though the cross-species transcriptomes are divergent, do you think that there are specific pain modalities, i.e. heat or cold pain, that are better conserved between species?
For that, I would refer to that PP2, PP1, PP2, or PP3, like this correspondence thing. So it seems like the TRPV1 and the TRPA1 population we found are more correspond to the PP1 population. So I didn't really go to detail, but for the human TRPV1, TRPA1, the 1.1 population, which we think innervating the skin, their expression level of piezo2 is very, very low. So even though we cannot just solely based on piezo2 to say they are not mechanical sensitive, but it seems like it's possible that V1 and A1 population are more for like temperature and a chemical pain sensation. While that PPK population, as we discussed in more lengthy, they have some expression of piezo2 and a mouse immediate, sorry, apologize for that. For mechanical pain sensation, and that seems to be conserved too. So I think in a large structure wise, if you think some fibers are more maybe responsible for this kind of like temperature and chemical pain sensation, and some maybe more for this mechanical pain sensation. That seems maybe, but based on the molecular profile, seems most of them should be polymodal. Even for the TRPM8 population, for the C-TRPM8 population, it's also expressed TRPV1 too. So that seems like a more dysregulated temperature sensation channel. So all I can say is, yeah, yes, maybe, but I think it needs much more, kind of like future functional characterization, as I said, maybe histological validation for the anatomic innervation and all those things. But I think the general framework should be held there. Yeah.
Thank you. Another question regarding the age and sex. Did the age and sex of the animals from which DRG was used, was that correlated to the age and sex of the human donors?
So we actually have only, as I showed at the very beginning, we have only sequenced 6 DRGs from 3 human donors. Yeah. 2 of those are females. One is about like more than 60, in their 60s, one is 58. And then we have another male sample, which is around like 23-ish. I don't exactly know the monkey data, like the age and sex, but for the mouse data here, we used for comparison for the guinea data. My understanding is they used 5 male, they used both male and females at the same age. So like around 2 months old. So so I would say, I mean, this sex and age thing is always a very interesting question, but at least for the human data right now, our sample size is too low for us to say, to draw any obvious conclusion. I mean, on the gross surface, as I showed, we don't really see obvious batch or donor difference. Or that's all I can say for now. Yeah.
Oh, thank you very much. For a second question from Will Renthal, he writes that the Ryba Davidson eLife paper suggested there might be human-specific DRG clusters, but the Yun et al. Paper recently published in Nature Communications did not observe this. Can you comment on whether or not you identified human-specific clusters, or if all cell types had mouse NHP equivalents and are mostly differentiated by marker genes?
So I would say, so this is something we, at the beginning, I try to emphasize a difference between the single nuclei sequencing and the single soma sequencing. I think they might contain somewhat different information. Part of those are because of the biological phenomenon, the nuclei and the soma, their transcript quality and quantity are different. But part of those could be caused by technology. The single nuclei sequencing, as I showed in the table, they use the 10x genomics platform, so that sequence depth is a little bit less, at least what has been published. So basically, the unique genes per single nuclei from those studies are about 1,000 and 2,000. But here, by using Smart-seq2, we actually get much more molecular unique transcripts. We get more than 9,000 per soma. So that's also a huge technical difference here, which might lead to this different. Actually, so this is an unpublished study, but we hope to be able to send out our manuscript and preprint maybe in the next, hopefully 1 or 2 weeks shortly. Actually, we did do, I didn't show the data here, but we did do analysis to correlate our data to the single nuclei sequencing data from the Davidson dataset. So there's some match, but most of those are kind of mismatch. So we think that the reason for that mismatch is both biological and technical, as I said.
Thank you very much. A question from Philippe Seguela from McGill. Great data, Wenqin. What are the implications of your results on the value of current preclinical models of pain in rodents?
So thanks for the question. I would say, please at least validate the expression pattern in the human data. So that's another thing. We actually have used a previous postdoc in Patrick's lab has already kind of makes our data searchable, kind of like a website saying you can type in the gene and you can see the expression pattern in these different 16 clusters. But we just cannot make it publicly available yet. So we will make it publicly available after our paper is accepted or published. So that's my suggestion. Please come to the data and at least the search to say, is your gene, is your target studied in using the model organism? Does it express in the right type of sensory neurons for translational study? I think this is very, very important. As the example I showed for the TGR5, if we don't look at it carefully, you wouldn't know, right? It's in the cellular glia cells, not the sensory neurons.
Thank you, and thank you for again noting the attendees about your upcoming paper in the submittal for submission. Actually, a follow-up to this is a very specific example to what you were just talking about. So NK1 receptor antagonists have failed in humans to relieve pain, but have worked well in murine models. So does your study provide an answer to this specific difference, species-specific difference?
So NK substance P expression I didn't show actually is fairly specific in the human DRG neurons and in the PEP nociceptors. I didn't show that, but solely based on that gene expression pattern of TEC1, you would think it will have some effect for the pain treatment because its expression pattern of TEC1 actually is relatively specific even in human DRG neurons. So then it's intriguing, right? I think the expression pattern is, as we are doing here, is probably the first pass, but then the human system is just very complicated. So the functional, there's just many other factors contributed to that, like why that translational field I cannot see. But yeah, but all I can see is the TEC1 expression pattern itself. Actually, it's quite specific in the pain nociceptors.
Thank you, and I'm going to reiterate the last comment from Will Rentals about beautiful work, and I'd like to reiterate that. And I also want to thank everybody. We're at the just slightly past the top of the hour, so I did want to thank everybody for having attended. And note that this talk has been recorded, and the link to it will be made available to everyone for those who want to go follow up and look back at the data. So again, I want to thank you, Dr. Luo, for presenting your work and generosity and time of sharing your new data and answering the questions. And again, thank you for the audience. And well, I'll see you next time in the next series. Thanks.
Thanks you all.
Bye-bye.
Bye.